we'll give it a minute or two here and see if anybody else hops on. But then we may just go ahead and get rolling. And because it'll be recorded, so people can watch it. Okay. There's people jumping on the Twitter and YouTube as well, so there is people watching. All right, so thank you everybody who's in here for joining us for our first graduate student seminar talk. So Christina is going to be talking today about PFAS, fate, and plant uptake from Biosolid-Amended soils, correct? And so Christina works with Dr. Lee in the agronomy department, and she is a PhD student. And with that, I will turn it over to Christina.
All right, let me just share my screen. Oh, can you host me again, please?
Yes, I can. There you go.
Thank you. Okay. Let's see if I can get the correct screen sharing real quick.
So you have the presenter view showing right now.
And now I fixed it, right?
Yep, you're good now.
All right, good. Thank you. All right, so today I'm going to talk about PFAS, fate, and plant uptake from biosolid-amended soils. I'm really excited to be sharing this with everyone in agronomy because it's a very interesting topic that has been very prominent in the news lately. And Dr. Linda Lee is my advisor in her research group, and a lot of the research I'm going to be discussing today has been research that was previously done by Dr. Rooney Kim Lascano. So I wanted to give a huge shout out and thank you for her paving the path for me to start this research. And with that, I'll get going. So what are PFAS? PFAS are a compound group called per and polyfluorinated alkyl substances. These compounds are present in a variety of media, including consumer products and other commercial products, as well as firefighting foams. They're used because they have some really great properties, including that they're nonstick. They can be water repellent, grease repellent, and stain resistant. So you'll find them in nonstick cookware, clothing, fire resistant fabrics, carpets, cosmetics, and like I mentioned before, firefighting foams. So exposure to PFAS can occur from a variety of routes, mainly if PFAS enter the environment or directly from consumer products. PFAS can enter the environment from industry, as well as use of firefighting foams, as well as once the compounds get into waste infrastructure, they can also be introduced to the environment. And then humans can be exposed directly from those consumer products, or when they ingest water, animal products, or crops that are grown in contaminated sources, as well as indoor inhalation and outdoor inhalation. So I mentioned before the widespread attention that PFAS have been getting. It's a national issue that has been identified by the Department of Defense, as well as the EPA, which has allocated a lot of funds to research on this topic. You can hear the term forever chemicals. It's a little bit easier to remember than the acronym. And so if you do some research there, you'll come up with a load of articles from different media sources. as well as some recent documentaries. There's The Devil We Know, which is available on Netflix. So if you guys are interested after this talk, you can watch that this weekend, or Dark Waters, which isn't on Netflix yet, but it's fairly recent. So I'm sure it'll come eventually, who knows. And Michigan has really paved the way on this targeting PFAS as far as regulations go, and testing across the state has been. So if you want to look into that, Michigan is very transparent as far as PFAS goes. But why are they a problem? Well, PFAS partition into the liver, kidney, and blood of humans as well as other animals. And they're found in pretty much all of our blood, which is particularly concerning. And you can see here, there's a graph of the concentrations in human blood and the nanogram per milliliter concentration over a variety of countries. So it doesn't necessarily discriminate too much. And in terms of when we're thinking about children, it's also in human milk. Now, granted, the concentrations here are in picograms per milliliter, but this is still an exposure route for a developing child. And this is kind of in contrast to some of the organic contaminants that are often based on a model of, I would say, lipophilicity, whereas the PFAS, like I mentioned, are proteinophilic. So some of the health impacts that are associated with PFAS, and I won't spend too long on this because there's a long list, are either to developing children in the womb or also to adults and children. So they can lead to thyroid disease, increased cholesterol levels, liver damage, kidney cancer, and testicular cancer. There's also some lower certainty health effects, which I won't go through. And in developing children, there can be delayed mammary gland development, reduced responses to vaccines, and lower birth weight. So when I'm speaking about PFAS, like I said, I'm speaking about a compound class. So the newest estimate in 2020 is that there's over 7,000 chemicals in this class, which can make regulation especially difficult, as well as analytical analyses and a variety of other factors. So the compound classes are divided into a couple categories. In this presentation, we're going to talk mostly about perfluoroalkyl acids and perfluoroalkyl acid precursors. So you can see here how they're split up. These are the perfluoroalkyl acid precursors, and these are the perfluoroalkyl acids. And of the perfluoroalkyl acids, the most studied are the perfluorosulfonic acids and the perfluorocarboxylic acids. So let me show you what those look like. This compound structure is a structure of PFOA. So you can see that it's got a carboxylic acid functional group. Here, this is the perfluoroalkyl, or the carboxylic acid functional group, and it has 8 carbons total, 7 of which are fluorinated. You can count those if you want. And here is PFAS. So it has a different functional group. This is a sulfonic acid functional group. It still has 8 carbons, but all 8 of them are fluorinated. And these are the 2 most studied, and you will also probably see them the most in the media if you look these up. Now, these compounds in these groups differ mainly based on their carbon chain length. So you can see here, it's very, very small. I'm sorry about that. That they can have a variety of chain lengths. And this can change their bioavailability as well as other factors. So when I'm talking about precursors, here are some good examples about precursors. This is a fluorotelmer alcohol. And it's a precursor because it can degrade to this perfluoroalkyl acid. In this case, it's PFOA, and it's got that carboxylic acid functional group like I was saying before. And over here, we have a sulfonamide, which can also degrade, but in this case, it could degrade into PFAS. Now, the thing is, once these compounds degrade from their precursors into these terminal metabolites, then they get pretty persistent. So they don't degrade much further from there, which means once they're in the environment, they stay there for the most part. So what I'm focusing on today is biosolids, and biosolids come from wastewater treatment. So how do PFAS get into wastewater? Well, they come from multiple sources, like I mentioned, the consumer products and other things that end up being treated through the wastewater treatment plant. Now, there's two routes for PFAS once they get into the wastewater treatment plant. They'll partition into the sludge, and they'll either end up in the water component, which is the effluent, or they'll end up in the biosolids. Now, as this process goes on, often it can seem that the concentrations increase from the influent to the effluent or biosolids, and that's because some of those precursors do degrade, but like I said, they will degrade into the perfluoroalkyl acids, which are persistent. So then what happens to the biosolids? Well, biosolid management depends on the locality, but 2.4 million tons were actually reported to be land applied in the US by the publicly owned treatment works. They're required to report to the EPA in 2019. So that was over half the biosolids. There were also a portion of biosolids in the US that were incinerated. That was about 16%, and about 22% were landfilled. So why are biosolids land applied? Well, there's a lot of benefits to land applying biosolids. One of them is that you're recycling waste, which economically makes sense, as well as providing plant nutrients. So these can be used as fertilizers. They have a great source of carbon and a great source of nitrogen, as well as improving soil health. There's also some studies that indicate that land applying them will reduce the carbon footprint as well. However, like I mentioned, PFAS do end up in these biosolids that can lead to a potential challenge of plant uptake and then leaching from these biosolids to water sources once they're land applied. So some of the studies that we have done on this at Purdue, and when I say we, like I mentioned before, most of this research was done by Dr. Rooney Kim Lascano, with some studies on occurrence. We did this occurrence study on commercially available biosolid products. So these are the types of things you could buy at Lowe's or Walmart to use on your garden, as well as some studies on treatment processes and plant uptake. And the plant uptake was completed in the greenhouse with soil that was amended with these products that I mentioned before. So in our studies, we looked at 17 perfluoroalkyl acids and we screened for perfluoroalkyl acid precursors. 13 of these perfluoroalkyl acids were perfluorocarboxylic acids and 4 were perfluorosulfonic acids. We looked at 18 commercially available fertilizers and growing media products. 11 of those were biosolid based and 7 of those were non-biosolid based. All of these products were obtained in 2014, except for milorganite, which we also obtained in 2014, 2016, and 2018. So here's a list of the available organic soil amendments that we were able to analyze. Our method included first freeze drying these samples, and then we added a mass labeled surrogate standard, which allowed for the analysis to take into account any losses that occurred in the process. They were then extracted with organic solvent and evaporated to dryness. Then we were able to reconstitute them with methanol. And at this point, they were either cleaned up and analyzed directly, or a total oxidizable precursor assay took place. Now, the total oxidizable precursor assay is a heat-activated persulfite treatment at initial pH values greater than 12, which can allow for the indirect quantification of perfluoroalkyl acid precursors that are not amendable to standard analysis. This can occur by oxidizing those precursors into the quantifiable degradation products. In this case, those are perfluorocarboxylic acids. So we either did the cleanup before or after this top assay. And then we transferred these samples to injection vials and analyzed them on our UPLC QTOF. So here is our data of the commercially available organic products. And the first thing you can see from this graph is that the Non-biosolid-based products had much lower concentrations than the biosolid-based products. You can also see that the range of PFAS in the biosolid-based products, and I'm talking here of the sum of the PFAS we analyzed, was between 30 and 185 micrograms per kilogram. Of these PFAS, the ones that had longer chains, greater than C6, were dominant in these samples. And then for the non-heat treated ones, they actually are more comparable than they look in this graph, because once we normalized it to the less than 2 millimeter fractions, the sample concentrations ranged more from 20 to 80 micrograms per kilogram. The reason we did this normalization is because we did some studies and we realized that the greater than 2 millimeter fraction had negligible perfluoroalkyl acid loads. So before I mentioned that we did the top assay before and after cleanup, and you can see here, this is a graph of the top assay. And we notice that after cleanup, the top assay is more efficient. And this is because the dissolved organic matter reduced the efficiency of the top assay. The other thing you can note is if you compare the top assay post cleanup to the pre-top assay, is that there are precursors in these samples. Now, once these precursors were broken down into the perfluorocarboxylic acids, the main one that was observed was PFHXA. So of these precursors, we were able to do a non-target analysis, and we confirmed a couple precursors at 2 levels. At level 1, this is a confirmed structure where we have a reference standard, so we're pretty much 100%. Level 2, we're pretty sure we have a library spectrum match, but we do not have a reference standard to make sure. So at a level 1, we found 3 sulfonamide containing PFAS, as well as 2 fluorotelomere sulfonates. At level 2, we were able to identify saprofluorotelomere phosphate diesters. We didn't look for these, but some precursors that are definitely of note in the literature and we will be interested in looking at in the future are sidechain fluorinated polymer surfactants. Recent literature by Letcher and Chu, both in 2017 and 2020, have indicated that these sidechain fluorinated polymer surfactants are present in high concentrations in biosolids. Their studies specifically analyzed S1 and S2, which are from Scotchgard fabric protectors, and they looked at Canadian biosolids. And in these biosolids, they noticed that the concentrations of just these 2 compounds were higher than the sum of the 22 perfluoroalkyl substances that are pretty comparable to what people normally study. So that's very significant. And in their other study of the biosolid-augmented soil, they found a similar trend that these compounds were at higher concentrations than the other PFAS. So this identifies that there's the potential for a continuous release reservoir of PFAS in these soils once they're amended with biosolids that have these sidechain fluorinated polymer surfactants. Now, like I mentioned, for the milorganite, we were able to analyze 3 years, and there was somewhat of a temporal change here where PFAS reduced by 80% between the 2014 and 2018 samples, and PFHXA, there was an observed 30% reduction between 2014 and 2018 samples. Originally, we considered that this might be due to regulatory shifts, but based on another sample that I'm going to discuss next, this might not necessarily be a case. So here you can see the comparison of 2 products that are from the same vendor. The product we analyzed in 2014 and the product that we analyzed most recently in 2019. Now our methods have changed since we analyzed the samples from 2014, but if you're comparing just the sum of the 17 perfluoroalkyl acids between the 2 years, what we notice is that in 2014, the concentration was 35 micrograms per kilogram, but in 2019, the concentration was 210 micrograms per kilogram. So that's a pretty big increase. Our total concentration in the 2019 product was 233 micrograms per kilogram. I want to point out this graph is on a log scale, so all of these PFAS listed below are not necessarily at concentrations that are that comparable. So that's important to note. Also, like I mentioned, our methods changed. One, we increased our list of PFAS to 67, and two, we changed our extraction procedure slightly to make sure that we're getting all the long chains in our analysis. So the final study I'm going to discuss today was on plant uptake of perfluoroalkyl acids. So this study was conducted at a greenhouse in Purdue, and we looked at two crops, a leafy vegetable kale and a root vegetable turnip. These crops were grown in Miracle-Gro potting mix, and they were amended with Milorganite, and we analyzed 17 perfluoroalkyl acids. So the Milorganite was amended with the Miracle-Gro potting mix at 3 application rates. The first one was 0, that was our control, and then 1, which is our recommended application rate. So this would be based on the optimal nitrogen that the plants would need. And then 4 times, and this rate was chosen because sometimes gardeners may choose to be heavy-handed. So it's not necessarily outrageous. It actually makes a lot of sense that the perception of people might be that if you add more, the plants will be healthier. So this was taken into consideration. And all of these crops were irrigated with nano-pure water. And this is because we didn't want to introduce another source of PFAS to the experiment. So the method involved separating the plant parts into their different components so we could compare, for example, the root to the rest of the plant. They were also freeze-dried, ground, and extracted with organic solvents. Then they were concentrated, and cleanup in this case occurred with a solid phase extraction. And then they were analyzed in our LCMS 8040 and our UPLC-Q TOF. So here are some of the plants that we harvested from that experiment. You can see the kale on top and the turnip on bottom. And one of the first things you can see from this picture is that the higher application rates of the biosolids did not harm the growth of the plant. In fact, it seemed to improve the growth of the plant, which makes sense because you're introducing that extra nitrogen, which is great for plant growth. However, when you compare the application rates between the turnips and the kale, with application rate increasing left to right, you can see that the PFAS concentration did increase with application rate. It increased both in the growing media as well as in the plant components. And this is true for both the turnip and the kale. Now, if you look at the different plant components, what you'll notice is the short chain PFAS tended to uptake more than any of the longer chain PFAS. So here you can see PFBA and PFHXA, and these are both perfluorocarboxylic acids, which is also of note. At the highest concentration in kale, you can see a similar trend, except in the root here, there's also PFDA. And the turnip, one of the interesting things of note was that the concentrations were higher in the peel than the bulb as well. So our conclusions from this project are that the soil amendments enhanced plant growth and plant health. And shorter chain PFAS tended to accumulate in plants more than the longer chain PFAS. And the longer chain PFAS tended to remain in the growing media, or they sorbed to the plant roots. The higher application rates also led to more accumulation in the growing media, and led to some increase in the uptake into the plant parts. So now I'm going to discuss the T on my recent literature review on plant uptake. So as I mentioned before, we think that precursor degradation and the potential can lead to the potential for a continuous release reservoir for plant roots to uptake PFAS. We think that the uptake of PFAS occurs mainly by plant roots. In most situations, however, if you're close to manufacturing, there might also be other routes of uptake. Transfer from the roots to the remainder of the plant have been identified as an active process that is impeded by the Casparian strip. In short chain PFAS, or very short chain PFAS, tend to have a couple more routes of uptake than the longer chain PFAS. Shorter chain PFAS translocate more readily from the roots to the remainder of the plant. This is partially due to their size and their bioavailability, which is mediated by their sorption dynamics. There were also some studies that showed that the plant root protein and lipid content can influence plant uptake. A very interesting study noted that the uptake varied between a few lettuce cultivars. So they looked at romaine, leaf, and head lettuce, and they identified leaf lettuce as the variety of lettuce that accumulated the lowest PFAS. I think they only looked at PFAS in this study, so it's quite limited, but it might lead to some interesting discussions in terms of the types of crops that you would want to grow on PFAS contaminated land. They also found that subcellular distribution between the cell walls, organelles, and water-soluble fractions depended on PFAS properties. I think this study only compared PFAS and PFOA. However, what they found was like PFOA tended to be in the more water-soluble fractions when compared to PFAS that preferred fractions of the plant that had higher lipid and protein content. Several studies reported phytotoxic effects of PFAS on plants. However, these phytotoxic effects, which included DNA injury, photosynthesis inhibition, and metabolic process impacts, generally occurred at really high concentrations, which would be higher than those you would consider in the field. There's also the possibility for a food chain level exposure. So although plant uptake doesn't necessarily occur at high concentrations in all the edible portions of plants, any of these plants that can be introduced are these plant components and residues that can be used in animal feed could lead to accumulation in animal products, which then serve as an exposure route for us. So given that PFAS are forever chemicals, shouldn't we just ban PFAS containing material from land application? Well, if we ban land application of biosolids, this in particular could place a heavy burden on public municipalities and taxpayers that support them, meaning that could even result in social disparities. Like we mentioned before, over half of the biosolids in the US are land applied, and that can bring in another source of revenue for these plants as well. As well as if we ban land application, something else has to happen to those biosolids. And we discussed before incineration and landfilling. However, with incineration, we're not sure that all of the PFAS actually will break down. So there's the ability for the PFAS to be introduced to the environment if we go this management direction. And if we landfill, there's also the possibility that PFAS will end up in the landfill leachate and then once again have to be treated by the wastewater treatment plants. Or if the landfill is not properly managed, that we could end up with a legacy problem we have to deal with farther down the road. So our recommendations are to regulate the non-essential uses of PFAS, for example, banning them in food packaging and use in carpets, which has already been done in certain areas, which also means that there's a demand for these products that don't have PFAS in them, which will make it easier for us to go down this road. And this will definitely reduce the PFAS loads in biosolids because it will reduce what's going to the water treatment plants. And as far as other sources go, we can pre-treat individual sources that have the highest levels before we start the long water treatment process. We can also look at specific points of contamination in the biosolid treatment processes. For example, looking at the equipment that's used, but also considering deposition from nearby industry and manufacturing facilities, as well as measuring the PFAS and identifying the precursors that are in biosolids prior to land application. This actually makes a lot of sense because if you're applying biosolids with very low concentrations of PFAS, you're gonna be land applying less PFAS than if you're applying biosolids that have higher concentrations. Another way to go about this would be taking those biosolids that have higher concentrations of PFAS and amending them with something else, like a lower concentration biosolid or another waste, like a compost. And then either those products will still have nitrogen in them, or you can amend them with other nitrogen sources, because all of this land application is driven by the amount of nitrogen that we're going to be wanting on these fields, because it's used similarly to fertilizer. Another option is to continue researching mitigation and treatment processes in the wastewater treatment process. And this is something that our lab is currently doing. We're also going to continue associated research to better understand PFAS plant uptake and risks, and to mitigate PFAS and biosolids in general. And with that, I want to thank my advisor, Dr. Linda Lee, as well as, once again, Dr. Rooney Kim Lascano, everyone listed here, and also Masa, our postdoc, that has contributed some research to this talk. And I actually have her Twitter here, so follow her. I think that's it. Richard, are you there?
Yeah, yes, I'm here. So hopefully people can hear me. If you have questions, please just unmute yourself and say who you are and ask your question.
Do we have any questions from our live stream?
No. We do not.
I can start off with a question. Hi, Christina. This is Charlotte. And I was curious, what were some of the most interesting findings to you as you've kind of been reviewing all of this past body of work that's been done with your lab?
Well, one of the things that really struck me, and I don't know if you could tell by the way I presented it, was those compounds that I introduced, the S1 and S2, and how they were at concentrations that were so much higher than the other PFAS that are the ones that were most commonly analyzed. So it's almost like those compounds weren't even being considered until now. So none of the studies included them. So there's a lot that needs to be done in regards to research on those compounds. And I'm hoping maybe even to do a little bit of research on those compounds specifically in my dissertation. The other thing about those specifically was I think that most of the research has shown that industrial sources lead to the highest concentrations, but of the S1 and S2 compounds, it was actually the treatment plants that received domestic waste. And this is because these are stain resistant, so they're used on carpets, etc. So consumer products contribute greatly to the concentrations of those. So that was kind of interesting and different than what had previously been discussed in the literature.
Thank you. It was also very interesting when you brought up, I grew up in Michigan personally, and I didn't realize that those same regulations weren't more widespread. Maybe that's because of some of the Great Lakes initiatives, but I know especially on things like detergents, they're pretty strict.
Yeah, everyone who's interested should definitely go to the website in Michigan. There's several states that have slowly gone on this train. I think it's mostly because they tend to act, I think, when there's not a lot of action on the federal level. However, there's been a lot of discussion about this in Congress, and so we may see some federal action. But once again, we're talking about 7,000 compounds. So unless you go through the argument of regulation with a compound class level, there's always going to be another compound to replace what's on the market. So that's something to consider.
There's a question in the chat for you, Christina.
So Adebo Cola is asking, would you propose using plants to reclaim PFAS from biosolid-amended soils? There are some people proposing this, and they do research on specific plants, not necessarily crops. Like there is a recent study that was done on a salix species, which is a type of willow. And they do uptake into these plants, but I wouldn't necessarily consider it the best way to go about it. Like it's interesting and phytoremediation is fun, and I want to like it, but I don't think that it's good enough to really tackle the problem. Hopefully I didn't offend anyone by saying that.
Questions from anybody else? Okay, hearing none, let's all give Christina a big virtual round of applause. We would have loved to be in person for this, but obviously due to what's going on, we can't. This will be our normal time slot, so we will have every week we'll have one student give a presentation, both on Zoom for the department and then on YouTube and Twitter as well for those outside of the department, so they can also join us there. Thank you everybody for participating, and hopefully we see you again in the future. And again, way to go, Christina.